Welcome back to the Deep Dive, the place where we turn complex research into essential, actionable knowledge. Okay, let's unpack this. We are diving into a really crucial piece of disease ecology today. It's based on a paper by Eloise Duval and colleagues titled, "When Does a Parasite Become a Disease? eDNA unravels complex host-pathogen dynamics across environmental stress gradients in wild salmonid populations."
Yeah, that title really nails the core challenge, doesn't it? We know infectious diseases out there in the wild, they pop up from this really delicate balance or imbalance between the host, the parasite itself, and whatever the environment's doing.
Right.
And for folks trying to manage fisheries or conserve species, the tough part is teasing apart, is it the number of animals, the biotic factors, or is it things like temperature, pollution, the abiotic stuff?
Especially in the wild where getting good data is just hard.
Exactly. It's incredibly difficult.
And the specific disease they're looking at here is a big one for European fish proliferative kidney disease, PKD. It really hits salmonids hard, especially brown trout, salmotruta.
Oh, absolutely. PKD is caused by this tiny parasite, a myxozoan called tetracapsuloides bryosalmona. Now, the parasite itself is often just there. It's endemic in many places. But when things get stressful, it triggers this massive infection inside the fish. Kidneys swell up, severe anemia.
And huge die-offs, right?
Yeah, huge. We're talking losses from 20%, sometimes right up to 100% in certain populations, wild and farmed. And high water temperature has always been sort of the prime suspect for flicking that switch.
Okay, so these researchers wanted to go deeper than just saying hot water, sick fish. They wanted to find the actual mechanisms, what turns parasite present into disease outbreak. And they tackled this with a huge study covering a whole region using some really advanced molecular tools.
That's right. They put together this integrated approach using environmental DNA, eDNA, across loads of sites in the Pyrenees and southern France. And this let them map out the whole picture, where the parasite is coming from, how much is in the water, and critically, are the fish actually getting sick?
Okay, let's break that down. First, the cast of characters and how they track them.
Mm.
The parasite's got that two-host life cycle, doesn't
It
it?
does, yeah. Needs two players. First, the main host, the definitive host, is this little filter-feeding creature called a bryozoan. Fredrotella sultana is the one in this area. The parasite reproduces inside the bryozoan and then releases these infectious bits into the water.
And then the trout comes along.
Right, the brown trout is the intermediate host. It picks up the infection, the parasite grows like crazy in its kidney, and then the fish excretes spores back out, continuing the cycle.
And the study was massive. 83 sites, 54 streams. They sampled in summer 2020.
Which is the key time, yeah. That warmer period is when PKD really tends to take off.
Okay, so methodology. How do they track the biotic stuff, the parasite and its hosts? They talk about propagal pressure. What's that?
Propagal pressure is basically how much infectious material is floating around. Think of it like the dose of parasite the fish are exposed to in the water. To measure this, they collected pretty large water samples, up to 12 liters per site, specifically for eDNA analysis.
And this wasn't just basic PCR, was it? They used something more advanced.
Yeah, this is where it gets quite sophisticated. They used multiplex droplet digital PCR, or ddPCR. The multiplex part is key. It means in one go, from that one water sample, they could measure the DNA concentration of the parasite, T. bryosalmona, the brown trout S. truta, and the bryozoan F. sultana, all at the same time.
Wow.
It gives you this amazing snapshot of who's where and roughly how abundant across the whole landscape.
Okay, so eDNA tells you what's in the water.
Yeah.
But how did they know if the fish were actually getting sick? I mean,
That-
83 sites, you can't exactly dissect fish everywhere.
No, exactly. So they used a really smart non-lethal approach, urine DNA or UDNA. They sampled juvenile trout, they're the most vulnerable, and measured how many parasite spores were in their urine.
Urine. How does that work?
Well, because the parasite multiplies like mad in the kidney right before the fish releases spores, the amount of parasite DNA in the urine directly reflects how bad the infection is inside the fish.
Oh, okay.
Yeah, previous work showed that high UDNA counts match up really well with how severe the kidney damage is and how likely the fish are to die. So it's a solid proxy for disease severity.
Got it. So eDNA for exposure risk in the water, UDNA for disease outcome in the fish.
Yeah.
And then they layered on all the environmental data, temp, water chemistry, land use, dams, all the potential stressors.
Precisely. They collected data on water temperature. The average was around 16.8 Celsius, but also conductivity, which can indicate nutrient levels, the percentage of farmland nearby, and whether there were dams upstream, plus how big they were.
Okay, let's get to the findings. Finding one, what drives the parasite's presence in the water itself? the propagule pressure.
Right. And this first part was, well, largely what you might expect, but the strength of the finding was impressive. Basically, where the parasite DNA showed up in the water was almost entirely down to biotic factors. So where its hosts were.
How clear was that signal in the data?
Very clear. Their statistical models showed that just knowing the eDNA levels of the trout and the bryozoan explained 51% over half of the variation in the parasite's eDNA levels in the water.
51%. And the environmental factors?
Hit the abiotic factors alone, they only explained about 5%, almost negligible in comparison.
So it really concerns that basic ecology, parasite needs its hosts.
Exactly. Local parasite numbers are dictated by host availability.
Now, it makes sense the bryozoan, the main host, drives it, but they also found a strong link with the trout DNA abundance in the water. That was a bit more surprising, wasn't it?
It was an interesting detail, yeah. It really highlights how the fish aren't just passive victims here. They're actively amplifying the parasite load. Once a trout gets infected, especially badly, it starts shedding spores in its urine, pumping more parasite material back into the river.
So the fish contribute significantly to the overall risk for other fish.
Definitely. They become part of the transmission cycle in a big way.
Were there any environmental factors that played a smaller role in where the parasite was found in the water?
There were a couple of secondary things. They saw a bit more parasite DNA where water conductivity was high. That might mean more nutrients, which could help the bryozoan colonies thrive.
Okay.
And interestingly, the presence of dams upstream also correlated positively with finding the parasite DNA in the water.
Dams. How so?
Well, dams tend to create slower-moving pond-like conditions, right? Lentic habitats. And those warmer, stiller waters can be really good for bryozoans. So dams might indirectly boost the parasite by helping its main host.
Okay, that sets up the big contrast really nicely. Finding one, biotic factors dictate where the parasite is. Now, finding two, what actually triggers the disease in the fish?
And here's where the story completely flips. When they looked at the parasite levels inside the fish, the uDNA measure of disease severity, it was primarily driven by abiotic environmental factors, stressors.
Oh, wow. Okay.
Oh.
So the amount of parasite in the water isn't the main driver of sickness.
It's necessary. Obviously, you need the parasite to get the disease, but it's not sufficient. The mere presence, even high levels, doesn't automatically mean sick fish.
That really is the core insight here, isn't it? That exposure and actual disease emergence are controlled by different things.
Absolutely. Quantitatively, the abiotic-only model explained 47% of the variation in how much parasite DNA was in the fish urine.
Almost half. And how much did the parasite level in the water explain, the eDNA?
Only 23%, much less influential than the environmental conditions.
So just measuring parasite DNA in the water is actually a pretty poor predictor of whether the fish population is in trouble.
That's exactly what this suggests. You need to know about the environmental context.
Okay, so what were the key environmental culprits making the trout sick? Temperature has to be high on the list.
Temperature was the big one, yeah. And it works in a couple of ways. First, warmer water speeds up the parasite's reproduction inside the fish kidney.
Makes the infection worse faster.
Right. But maybe more importantly, brown trout are cold water fish. When the water warms up, their bodies get stressed, their metabolism goes up, they need more oxygen.
That takes energy.
Exactly. They have to divert resources just to cope with the heat, which means less energy is available for their immune system. They become physiologically stressed and immunologically weaker.
So the warmth weakens their defenses, letting the parasite run rampant.
Pretty much. It creates this vulnerability that allows the infection to become full-blown PKD.
It's like they have to choose between breathing and fighting infection. What about agriculture? That also popped up.
It did. Sites downstream from areas with more agricultural land had higher levels of parasite infection in the fish. The likely link there is water quality. Runoff from farms can mean more nutrients, maybe pesticides or other pollutants.
Stuff that's just generally bad for fish health.
Yeah, it adds another layer of stress, further compromises their physiology, and makes it harder for them to fight off the parasite, even if it's present at only moderate levels.
Okay, and then there's the dams again. This was weird, right? Dams increase parasite in the water, but decrease disease in the fish. How does that work?
Yeah, that one's genuinely paradoxical and really interesting. It shows how complex these effects can be. So remember, dams seem to help the bryozoan host, leading to more parasite spores in the water that's finding one, higher exposure risk.
Right.
But for finding two, the presence of dams was linked to lower parasite loads in the fish urine downstream.
Why?
Well, a couple of possibilities. Dams can sometimes moderate downstream temperatures, maybe preventing those really extreme heat spikes that cause the trout's immune system to crash.
Ah, like a thermal buffer.
Could be. Or dams physically block fish movement. Maybe they prevent the most susceptible young fish from moving into the most highly infectious zones just below the dam where bryozoans might be densest. So it's this trade-off. Dams might increase the overall parasite load available, more spores from bryozoans, but simultaneously reduce the fish's vulnerability in that specific downstream reach, perhaps through temperature effects or movement barriers.
Fascinating. So the take-home message seems crystal clear. High parasite load is the background risk, but it's the local human-link stressors, warmth, maybe pollution from farming, that actually pull the trigger and cause the disease by weakening the host.
That's the essence of it. And what does this mean practically? Well, for managers, just mapping where the parasite lives isn't enough to predict disease outbreaks. You have to understand host vulnerability, and that's primarily driven by environmental stress.
So this eDNA plus environmental data framework they developed, it's actually a usable tool for managers on the ground.
Absolutely. It gives them a way to map risk more intelligently. They can use eDNA to see where the parasite is, then overlay things like temperature data or land use maps to identify which of those locations are likely hotspots for actual disease emergence.
They gave an example, didn't they? The Nesty River.
They did. They found parasite eDNA all along this river. But when they looked at the fish using eDNA, infections were only really happening at the lower sites.
Why the difference?
Presumably, those lower sites were warmer or maybe had more agricultural influence. But crucially, one of the uppermost sites had parasite DNA in the water. But the fish were clean, no uDNA detected.
Ah, so that's a site to watch.
Exactly. That site becomes a priority for monitoring. The parasite's there, the risk is present, but the fish are currently coping because the local environment is still relatively healthy. It's an early warning system.
Which implies conservation efforts should maybe shift focus a bit. Not just fighting the parasite, but bolstering the host's defenses.
Precisely. Trying to eradicate an endemic parasite is often futile, but managing the environment is feasible. Actions that target those key abiotic stressors, like reducing agricultural runoff, controlling nutrient inputs, maybe finding ways to mitigate temperature increases through riparian planting or flow management, those things strengthen the fish's natural resilience.
Making them better able to coexist with the parasite without getting wiped out.
That's the goal. Healthy environment, healthy fish, even if the parasite is still around.
Now, they were careful to point out some limitations too, weren't they, for future research?
Yeah, absolutely. They stress that this is a large-scale correlational study. It shows strong patterns, but to really pin down the cause and effect mechanisms, you'd need more localized experimental work.
Like actually manipulating temperature or nutrients in controlled settings.
Exactly. Plus, there's a technical challenge with eDNA right now. It can't distinguish between the infectious spores coming from the bryozoan host and the non-infectious spores being shed by already infected fish in their urine.
Ah, so the eDNA signal's a mix of both.
It is, and being able to separate those two sources would make the risk prediction models even more powerful. That's a next step for the technology.
So to wrap it up, this research really hammers home a vital point in modern disease ecology. Biotic factors, the hosts, determine where the parasite can live, but it's the abiotic factors, environmental stress, often human cause, that determine where the parasite actually kills.
Perfectly put. And it leaves us, and you, the listener, with a really important question to ponder, I think, given that the environment can be a bigger predictor of disease severity than the pathogen itself. What are those subtle, maybe overlooked environmental stressors acting in your local ecosystems? The gradual warming of a stream, the slow creep of nutrient runoff, things that might be quietly weakening wildlife resilience right under our noses.